Hello aspirant. So in this particular module, we are going to see why bioequivalence is important. What brings bioequivalence is more critical during the drug development or during the generic formulation development. How the bioequivalence comes into the critical process of the drug development. So these are the things we are going to see. So what is bioequivalence? So as we explained earlier, bioequivalence is comparison of the systemic exposure profile of the test product with the reference product. What is systemic exposure profile? So here the systemic exposure profile is nothing but the bioavailability of the drug molecule present in the drug product. So once you consume the drug product, the drug will be released from the drug product inside the systemic circulation, and then the bioavailability comes into the picture. So what is bioavailability? Here bioavailability is defined as the rate and extent to which the active ingredient or active moiety is absorbed from the drug product and becomes available at the site of action. So when you consume a drug product, it goes into the gastrointestinal tract and the drug will be released from the drug product, get absorbed and goes into the systemic circulations. This is a typical profile for a oral dosage form. So the rate at which the drug is getting absorbed from the gastrointestinal tract into the systemic circulation and the extent at which the drug is getting absorbed, the rate and extent both are being called as bioavailability. So this rate and extent will be compared for the test product and the reference product. This is called bioequivalence. So in a pharmacokinetic profile, the rate is denoted by the Cmax and Tmax, concentration maximum and the time to reach the concentration maximum, and extent is denoted by the area under the curve. So the comparison will be happening for the Cmax and the AUC for the particular drug product. How the bioavailability evolved. So here we are explaining how the drug response along with the drug concentration is evolved into the bioavailability. So the response is related to drug dose. How? So you might have seen in the hospitals or during the drug treatment. For example, paracetamol 500 mg you are giving to a patient and the fever is not reduced or the pain is not reduced, you are going to increase the dose sometimes. So basically that is called the drug dose related response, where you increase the dose sometimes without compromising the safety to achieve a specific response or reduction of fever or reduction of pain. So the response is basically related to drug dose, and the response is better related to drug concentration at the site of action. So the site of action, how much drug concentration is available is important for the response. That is the reduction of fever or the reduction of pain, for example. Then the plasma drug concentration reflects the drug concentration at the site of action. That is called systemic exposure. So once you consume a drug product and it get absorbed from the gastrointestinal tract and goes into the systemic circulation where the drug concentration is measured. So the plasma drug concentration reflects the drug concentration, the site of action is considered. So then the bioavailability means the drug available in the plasma, that is the plasma concentration profile of the drug product will be called as the bioavailability. So when we compare this plasma concentration profile of a drug is called bioequivalence for the two formulations. So basically what we are trying to say is that the response, that is the activity of the drug, for example the reduction of fever by the paracetamol, the response is related to drug dose. The drug response is better related to drug concentration at the site of action. Then the plasma drug concentration reflects the drug concentration at the site of action. So here the bioavailability defines the plasma concentration profile. Then we compare the plasma concentration profile for the assessment of bioequivalence of two formulations. So based on these fundamentals and scientific principles, we define the bioequivalence. Bioequivalence means the absence of a significant difference in the rate and extent to which the active ingredient or active moiety in pharmaceutical equivalent or pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. So here, the active moiety is the drug, then the pharmaceutical equivalent or pharmaceutical alternative are the dosage forms, and site of drug action is the drug for which the specifically administered, and the same molar dose means the dose of the drug should be same, and similar conditions will be maintained at the bioequivalence facility by appropriate protocol design and measures. So this is how the bioequivalence is defined, and all the aspects of the bioequivalence will be maintained during the bioequivalence study. So we have seen what is bioequivalence, defined bioequivalence. Now we are seeing here what is pharmaceutical equivalence, which we have seen in the earlier module also. So in a detailed manner, what is pharmaceutical equivalent? Pharmaceutical equivalent products are the same product of the reference product which contain identical amounts of identical API. That is, the salt form of the API also should be same. And then there can be a different inactive ingredient. Need not to be the same amount of or same qualitative composition of the ingredients. And it meets the identical compendial or other applicable standard for identity, strength, quality, and purity, including potency, and wherever applicable, the content uniformity, disintegration time, and dissolution rates are similar. So basically, pharmaceutical equivalent is nothing but if you have paracetamol of 500 mg, the equivalent product also will have paracetamol 500 mg in the same salt form, and the inactive ingredients can be different, but it meets the identical compendial or other applicable standards like identity, strength, purity, etc. Here we are going to see what is pharmaceutical alternative. So pharmaceutical alternative contain the identical therapeutic moiety or its precursor, but not necessarily in the same amount or dosage form or the same salt or ester. So what we are trying to say here. So you can take a perfect example of diclofenac. So diclofenac is available in the market in different salt dosage forms. I have seen diclofenac sodium and diclofenac diethylamine esters. So if you go and see the label of these dosage forms, you can see that each gram of diclofenac diethylamine gel contains this much amount of diclofenac, or each gram of diclofenac sodium contains this much amount of diclofenac. Eventually, if you look at the active moiety of diclofenac remains same in both the drug product. Probably the diclofenac diethylamine will be more and diclofenac sodium will be less, but the amount of diclofenac in both the salts will remain same. So this is how the alternatives are surfaced. So the pharmaceutical alternative is the same therapeutic moiety, that is nothing but the diclofenac, but the precursor or salt forms are different like diethylamine ester or the sodium. So here the diethylamine salt or the diclofenac sodium salt, each drug product in forms of gel or in terms of anything else meets their own respective competitor or applicable standard for identity, strength, quality, purity, etc. So now we need to understand why bioequivalence is required. So there are important three purposes of bioequivalence in the pharmaceutical industry. So number one and most renowned purpose is that to obtain marketing approval for generic products in regulatory and semi-regulatory markets like United States, Europe, Australia, Canada, and also to some extent in semi-regulated markets like Brazil, South Africa, India, Russia, China, etc. So basically, bioequivalence 99% of the bioequivalence studies are carried out for the marketing approval of the generic products. And then the other purposes includes the clinical trial product and to be marketed product. So what is clinical trial product and to be marketed product? The bioequivalence studies sometimes will be carried out during the phase trials of the new chemical entity where you manufacture already a clinical trial product and you have completed the phase 1, phase 2, phase 3 clinical trials. Now, after completion of the phase 3 trials, you have submitted the NDA for the marketing approval and you got the marketing authorization for the particular new chemical entity. Now you are going to market the product. So for that, you are going to make the commercial batches, not the clinical trial batches you will be marketing. So there should be one study as per the prescription by the regulatory agencies to be conducted to match the clinical trial product with the to be marketed product. So there will be a clinical trial product where you are done already established the pharmacokinetics, and now you will be having the commercial scale product, and both products will be administered to healthy subjects and the bioequivalence will be proved. So this is one important purpose during the new chemical entity development or new dosage form development, the bioequivalence will be carried out. And finally, sometimes when you are going to change the manufacturing facility, for example, your product manufacturing in United States, and for some reasons you wanted to change the manufacturing to the India. So in that case, you have to do some bioequivalent studies for both the products which is manufactured in United States and to be manufactured in India. So sometimes this can be bio waiver will be applicable provided you are generating some in vitro in vivo correlation data, etc. But most of the cases, the bioequivalence need to be proved if you are changing the manufacturing plants. So these are the three important purpose we do the bioequivalence studies in the pharmaceutical development. We have seen that bioequivalence studies are being carried out for generic drug product development. But why a generic product is required? So that is very important to understand. We have already seen that what is a generic product, which is identical or bioequivalent to the branded product, which is already marketed. So now the generic product is safe and effective alternative for the branded products. So now what is the use of generic product? So what a generic product can do is it can help both consumers and the government to reduce the cost of the prescription drug. So for example, the drug cost in the United States is 10 times higher than the cost in India, because the generic drug product cost, the production cost is much lower than the cost which is prescribed for the branded drugs in the United States. So basically, the generic drug products are used to reduce the cost of the prescription drug, and there can be several generic players which can compete and reduce the cost. So branded product are generally being marketed by only one person who discovered the drug product, but there can be multiple generic product players who can reduce the cost of the drug product and can market the product. It will help the consumers and the government. When comparing a drug discovery process and a generic drug product development, what difference the bioequivalence can make? That is what very important to understand here. So basically, this table represents both drug discovery process and the other side it present the generic drug product development. So in case of drug discovery process, we are seeing the different steps including chemistry, manufacturing, controls, labeling, testing. Then it comes to the preclinical studies like animal studies, then the clinical studies like phase studies, phase 1, 2, 3, 4 studies, then the bioavailability also part of that. But in case of generic drug product development, you have all the same thing what you have in the drug discovery process, like chemistry, manufacturing, controls, labeling, testing. But these preclinical, clinical studies like phase studies and pharmacokinetic studies are replaced by single step of bioequivalence study, where the safety and efficacy of the product is proven by the bioequivalence studies. So this is how the difference of bioequivalence can make into the drug product development, and it can be different for the drug discovery process and the generic drug development process. This particular slide represents the global generic market, how big it is. The global generic market in case of 2001 was 47 billions, and in case of 2006 it was 67 billions, and of late of 2020 it has been crossed the $100 billion value, and the patent expiry of highly profited branded drugs are estimated to be 37 billions in case of 2015, and the regional generic market in the US is 32% of the total market, and in Europe it is 44%, and in Japan it was 10%. That means 32% of the US market is comprised of the generic drugs. and in Europe it is 44%, and Japan it is 10%, where in India it is more than 95%. So this particular slide represents the global value of generic market with respect to different continents, including US, Canada, Latin America, Western Europe, Eastern Europe, China, Japan, and Africa. So this particular slide is showing the contribution of India to the global generic market. So Indian share of the global generic market is very high, and India is an important player in world generic pharmaceutical products. So the generic product contribution by India by different companies in present in India includes Lupin, Glenmark, Sun Pharma, Dr. Reddy's. In terms of market share is presented in this slide. So it is more than 30% of the generic drug products in the US is being supplied by Indian generic players. This slide showing some of the regulatory agencies across the globe in different countries, including India, USA, Canada, Europe, South Africa, Brazil, and Australia. So, aspirant, in this module, we have seen why bioequivalence is required, why generic product is required, what is the importance of bioequivalence, and what is the importance of generic product, and how India is positioned in the global generic market, what is the market share, and what is the importance of generic product presence in each and every country. So now we are going to see more detailed scientific principles and flow of activities of bioequivalence studies in the upcoming modules. Thank you.